Welcome all today. We're glad you're here and glad that you're participating with us in this virtual student symposium for code. And with that, I'm going to hand it over to Jeremy and let him get started.
All right. Can everybody hear me fine? I guess Michelle will be the only one that can nod, or I can know. All right. So I have the Q&A pulled open, the chat open, the participant list pulled open on my other screen. So hopefully if anybody pings anything throughout this, don't hesitate throughout to ask questions. I'll try to flow with them as I go through the presentation. All right. The screen share is working okay, Michelle? Everything's
Yes,
cool?
it's working great.
All right, so this is a new talk. These are relatively new slides that I've never presented before. So you guys get the first glimpse at them. But the goal of this is instead of being so much of the research successes we've had and things like that, this is more of how do we build more digital platforms for remote-based projects, especially in this COVID era. And you'll see that it actually is a double meaning. Because the COVID itself creates a lot of opportunity to do new research projects that are much more timely, as you'll see through these slides. So this first picture, I really love seeing this picture. This was my lab last summer, not this summer where we actually had nobody on site. But in this picture is a lot of undergrads. And that's really what my lab operates on, is undergrads and medical students. So we've got Olivia and Xavier here on the edges that are from Walsh University in Ohio. We've got Leah, and I'm sure several of you guys know Leah. She was a code student that came up and had a very successful internship. We have a couple undergrads from a community college that's local, and then a gap year student who's now off in medical school, who was very successful in his 1 year working with our group, doing a lot of these types of projects. So, it just goes to show that we ourselves are composed of a lot of undergrads in our group to try to bring these. And the goal is to take what we learn in our group and try to expand it to all of you guys and your guys' teams as well. So the goal really is to spark some interest in building a digital-based research platform and giving examples of projects and tools, and especially some of the newer tools that I've stumbled across in the last few weeks that I know Cindy's group and UNA have been asking, how do we make this easier? Hopefully I have some answers on these slides that will make your guys' life a little easier in doing variant polls and stuff like that. So I really wanted to start off with a timeline though. This to me just goes to show how fast things really can move sometimes, and the amazingness of this session and the posters that are online. And I look at the dozens of posters, and it makes me smile realizing how fast this program has grown. In 2013, when I started my postdoc with Howard in the Jacob Lab at the Medical College of Wisconsin, we really started postulating these types of programs. Really a lot with MSOE, and I know Michelle and others know MSOE's team a little bit, the Milwaukee School of Engineering. But this was the start, Nick Volker, and how do we make what that 1 in a billion story into research projects and educational material. And from there, we started launching some research partnerships. Walsh University, that you'll see quite a bit in the slides, in Ohio, and St. Leo, which is a private university down in Florida outside of Tampa, where we really got things kick-started at first. But it really was in 2015 when I was able to join Hudson Alpha and met, Dr. Lamb and Michelle and the education team that had really got this stuff kick-started. And starting with Athens State, I know Dr. Klein is on the line, and what we were able to start there, and Wallace State, a lot through Casey and her bringing Wallace State into it, and then really getting UNA, and these 3 schools really are what push so much of the code success forward to where it is now. And so in 2017, that was really the first funding, and when this got really kick-started, that was also the same year, and Casey's on the line here. So that's the first actual, code-ish based student before code even existed that really had a co-author slot on one of our publications and contributed data into a publication. And that was when we kind of said, hey, you know what, this isn't just, fun research. This really can be publishable. And as you guys know, in 2018, I moved up to Grand Rapids to start my own lab up. which thank goodness we've still been able to work together, and I still have so many wonderful contacts down south. It's cold up here, so sometimes it is nice to think about how nice it is down south. And we started expanding a lot of these tools into our Western Michigan school systems, and you'll see some of the slides on some of the ongoing projects that we're starting to kickstart there that I hope can give some ideas to schools down in Alabama. 2019 was actually probably one of our biggest moments for the code program. And again, Sarah, this is so much at the back of Athens State and what we were able to get going there. This was our first author paper from a undergrad student in these programs. And so Joy, who I know just recently is finishing up her master's degree and got a position in academia, in industry on a genomics-based company, and her work in multiple sclerosis, where this is something she said, I want to work on this. We said, all right, let's put some tools in your hand. She developed the project. She put everything in place. She helped write the paper. She got it all the way through. She went through all those struggles, and now she has a publication to show from it. 2020, we've had a couple papers now from students this year, and the one I really am going to highlight throughout this talk is going to be this stuff on COVID-19. And the students in red here are undergrad students from multiple institutes, and the faculty in blue here are those that have contributed, more of the mentoring and a lot of the logistics behind this project, and how we can all come together over multiple institutes to really build bigger research platforms. That was published in about a 4-month window from start to completion to publication in a very high impact good journal, JBC. It just goes to show that this is not fun research. This is real research. We're doing stuff that is publishable in high impact journals. So the outline of the rest of the slides, we're going to start off with some genetic variants and hopefully give you guys some tools. Many of you guys know, some new, just a couple slides on modeling and dynamics. And then I really wanted to explore some new project ideas and pilots and start getting some people thinking about what we could also do outside of just modeling and dynamics of proteins. And then give that COVID-19 example at the end of a bigger initiative that we can start pulling together to do. So most of you guys have seen this figure if you've been around me. It's a figure we made for a research article. And I apologize, my dog is right next to me, and so he may bark through this. There's no slides on him today, so he has to be feisty some other way. This slide really just shows the timing of DNA discoveries. And so, we've gone through 100+ years of insights and discoveries, but really in the past 17, 18 years is when so much of this has come into a new level, where groups like the Stanford Genetics that now has become Hudson Alpha were able to sequence the original genome. And from that turning point, which is a histone octamer in the turn of DNA, from that turning point, we're really able to start seeing how much genomics can amplify and how fast we can start sequencing thousands of patients and individuals, whether healthy or sick, all the way into hundreds of thousands of individuals, which means millions to hundreds of millions of genomic variants that need to be characterized and classified. And it creates so many research projects that we're never really going to run out of things to be able to look at or see. And so, some examples, tools like Nomad that many of the students hopefully have been starting to get some access to and looking at, where we know it's not just sequencing a few individuals, we're sequencing hundreds of thousands and of very diverse populations, which means you can also build projects looking at genetic variants that are specific to certain populations. Evolutionary biology. You'll see later on, I'll show a slide on the cystic fibrosis stuff, where it was actually done by an anthropology undergraduate student who was interested in the evolutionary behaviors of it, which then stemmed into a larger project. But even things like anthropology with genomics now, there's large-scale initiatives that these code-based tools can really, really help answer questions to. And so, with Nomad and Nomad-based tools, this is just one gene you can throw into the search tool, get that gene, get to a table of variants, you can extract them, filter them. That gives you the ability to now start extracting the information in a much easier format for large-scale population genomics. But what most of the students are really interested in, what most people really care about in genetics, is that of disease, right? And so we heavily rely on tools like ClinVar, which are those tools that are going to list variants that are linked to clinical pathologies, which are going to give you connections to genes and different conditions. This is unpublished data that I'm currently working on for a massive review article that we've been asked to put together. It's more of a book than a review article. But we've been really trying to go through large-scale data filtering of the entire ClinVar database. And this really highlights the need of these programs, the need of the number of research projects. If you take all of the variants in ClinVar, about 40% of them are in uncertain significance realm, which is, you know, way larger than that of the pathogenic and likely pathogenic classifications. And so you're talking, just alone, if you were to try to build a project on every single variant that's a VUS within ClinVar, you're talking over 300,000 projects. So sheer mass of these challenging variants. And we know that most of these VUS, VUS being variants of uncertain significance, are mostly missense variants. And so the largest portion of variants within ClinVar are missense. And when you look at the distribution of pathogenic or VUS over the different classifications of variants, we can see the pathogenic variants are actually more likely to be those of loss of function, right? Frame shift, nonsense, splice-based alterations. That is where our more confident calls come in pathogenicity calls. Whereas the missense variants actually make a much smaller portion of those that are pathogenic, but it makes up almost the entire portion of variants of uncertain significance, right? And that just goes to show, if you lose half of your protein, obviously it's going to impact the protein, and it's easy to say how that impacts it. Subtle changes, flipping one amino acid from one to another, they're much more complex, and we didn't really have tools up until a few years ago to really be making sense of these. And that's just the reason and need for the code-based programs and these research projects is the majority of missense variants that we see in genomes, we have no idea how they impact the proteins. And many of the variants you can look at within ClinVar, you'll be one of the first people to look at those variants because there's just so many of them now that keeping up with that is challenging. So, and that means, again, so many protein projects that focus on the protein. This isn't even getting into the realm of how variants that control gene expression and the regulation of them impact, where clinically we don't even know what those variants do or what they are, which will be the next wave to come in the future of starting to understand how do we make sense of non-coding variants, which you'll see a couple slides on later on. So one of the tools that I've not actually passed on to you guys yet, because I had needed some time to actually vet this tool and start making sense of it, is this tool called Simple ClinVar. And especially for the UNA group, I know you guys have been asking for how do you get better ClinVar data pulls. This is the answer. And so Simple ClinVar is a tool that the Broad Institute has put out. It was a publication last year, but the database really has gotten launched. You can see in about July this past summer. It is a searchable tool. You can throw your gene, throw your protein, throw your disease or disorder in here into the search tool and get clickable boxes that you can say, I only want pathogenic variants for something like epilepsy. So I threw epilepsy in here, and it'll give you nice, pretty tables of the breakdown. So how many are missense, stop, and you can see I obviously only have this turned on for pathogenic. But then you can also click and you can get the variants in a table. You can click the gene tab, and the gene tab will give you a very nice spit out of the genes that are actually associated with that term most often. And so, for example, in epilepsy, I can see SCN1A is probably the gene that I want to maybe build a project on if I'm focusing on epilepsy. Or something like GRIN2E, which I saw multiple GRIN2E posters from UNA. We have a publication in review right now on GRIN2A and a big database of characterizing variants. Right, you can start seeing which variants, hey, I might want to prioritize and look at in a characterization project. So this is a really nice tool if a student comes in and says, I'm interested in disease X. Say, throw disease X into Simple ClinVar and go to the gene page and start telling me which are the top genes in the rare disease space that might be worthwhile to go after and build projects for. You can also click on each of the genes, and it's going to come up with a nice, scrollable table with a download CSV file. And this is really what I think you guys have been looking for. It also not just has that, but it's going to give you really nice simple annotations of the reference, the change, the position, and then since you guys are working with Hudson Alpha, CAD scores, which are included in here, which are a tool that Hudson Alpha has been a co-developer of. That the higher this score is, it means the more impactful the variant. So just quickly, looking at these variants for SCN1A, you can see, hey, there's a VUS right here that's a missense that has a high predicted CAD score. So that might be a variant that I want to prioritize and build a model of SCN1A embedded into a membrane, look at dynamics. We have been systematically analyzing ClinVar, like I mentioned, as part of a review article we're putting together. And we know that there's a heavy distribution of genes with pathogenic and VUS calls, that it tends to be only a few variants for most of the genes that we end up seeing in it. But there are a few genes that have a lot. So there are 35 genes that have more than 1,000 VUS annotated to them, whereas there's only 7 pathogenic genes that have more than 1,000 pathogenic variants annotated to them. And this chart right here kind of shows you the distribution of the genes, which, when you look at this, you can see genes like BRCA1 and 2 that Hudson Alpha has spent a lot of time working on in the cancer initiatives. You can see essentially they have a lot of pathogenic, but they also have a lot of VUS calls to them. Genes like ATM and APC, a lot of these that are more cancer-based, have a little bit higher VUS numbers than that of pathogenic. Genes like Titin, which TTN, which is the largest gene in the genome, has a very large number of VUS. And this is one of those genes that is very tough to make sense of variants because it's so big. That makes it a very big challenge to do work with. And then there are a lot of the rare diseases. So you see SCN1A, right? A lot of SCN1A variants are more pathogenic, and there are only a few VUS relative to the frequency of pathogenics. The other set of genes that students can actively work on and develop a lot of very useful tools that are easier to publish is focusing on things like the ACMG59. And the ACMG59 are 59 genes where the mutations are very well connected to, so mutations in that gene have a high probability of being a causal disorder with a very clear defined list of phenotypes. And those 59 genes, there is some actionable intervention to it. So if I know these variants, I can change something in clinical management. And so making sense of VUS in the ACMG59 has a higher priority. And you can see the ACMG59 genes are really interconnected. You've got certain pathways that are more connected to the cancer mechanisms, like your APCs and your BRCA1s and 2s. And then there are some that are more cardiovascular. There's actually quite a large number of cardiovascular genes. So building out programs in cardiovascular variants. And then there's a portion that are actually neural as well, but neural tends to not have as much actionable biology on it. That is starting to change and shift actually in the recent year or two, that you're going to see more and more genes from neurobiology drifting into actionable, especially in areas like epilepsy. And then you've got areas of cancer, which normally when I first throw out options of projects for students to work on, the first place they normally go to is cancer, right? Well, I want to work on cancer, I want to look at cancer. And so tools like Cosmic that many of you guys are familiar with, where you can search genes, cancer types, and get lists of variants. Now moving into the project itself, right? This is, we oftentimes in code and the projects with our group and our lab, we oftentimes focus on tools like Yosara, yet another scientific artificial reality applicator, because of their multiple integrations, that there are so many tools integrated into a single platform. There are many other platforms out there, but I have to say most of our training sets that we've developed, and I know Michelle's group and Neil's group have really been pushing forward with Yosara as well because of its power. And so, my group continues to put out reference resources as well, videos on the basics of Yosara that you can access through our website or through our YouTube channel, Molecular Modeling, all one word in YouTube. So there's videos in homology modeling, basics of Yosara. Hopefully there'll be another wave of videos dumping here in the next few months as I play catch up from summer-based research. And when you can't get homology models, we typically will refer people over to tools like iTassr. So if you try to generate a model in Yasara and it doesn't work, that's where we'll kick you off to another tool like iTassr, which doesn't give you as good of models, but it gives you models in cases where there are no structural templates to be able to be used. And so it gives you an ability that if you can't get something from Yasara for the region of a protein you're interested in, you can go into tools like iTassr, which has a submission system to it. For the dynamics of proteins, this is where we move that next level, right? We don't want to just look at static stuff, but we really want to look at how a protein, you get that model, and we can look at more complex environments. So this is the CFTR protein in cystic fibrosis. Right? You can actually embed that protein in a membrane. If your guys' schools start doing membrane embeddings and you're running into difficulty, don't hesitate to reach out and ask. I was on a meeting with St. Leo down in Florida, who's been working on this stuff for like 8 years, and had to help troubleshoot a project for them, because this can be tough, but it moves your simulations and modeling into a whole nother realm, actually having a membrane and water built on both sides. And then that ability to track the movement of whether it's the whole protein or each individual amino acid. And some of you guys I know have started getting into the dynamics cross-correlation matrix data. This is really one of the most powerful things about the data you can generate from a simulation, where we track every one amino acid's correlation and its movement trajectory with any other amino acid. And what that gives us the power to do is say something like, hey, this variant in red here, actually in three-dimensional movement is tracking along with maybe this variant here. And so we know something like Delta 508 is behaving very similar to another variant that might be a VUS. And so we can say, hey, this VUS actually behaves and looks just like that of a known pathogenic variant. And we can build massive databases. Once we have this matrix created of the protein normal, you can start screening that through for thousands or hundreds of variants that's already pre-computed data. And so that's what we've been doing in this work in cystic fibrosis, is running the base proteins and then scaling and trying to assess every variant within CFTR and how that amino acid looks in it and how the change may impact it. And I highlight, this is a paper that was published in an Impact4 journal, very good genetics journal. And Morgan is an undergrad anthropology student who just graduated. So we took a very diverse field from what we normally work in and made a really cool research paper that got published, actually in one iteration of very minor reviews. Probably the best revision process of a paper I've ever had, which I think speaks to how well Morgan can write. So the last couple things I want to throw out before we move into the SARS example are some of the newer projects and pilot expansions that I've been thinking about. And so, UNA, I think is the first one I can highlight here, which is trying to build up our 3D printing workflow. And this was really when Leah came into my group over the summer, last summer, was really her push. She really wanted to go into genetic counseling and figure out how we make better educational tools. This has turned into, I think, about 20 projects for my lab now in 3D printing educational materials for various clinics all across the US. So it started from one undergrad's motivated ambition and turned into a huge amount of stuff into my group's research program. And the whole idea is to take those models, Sarah, you're on here, so looking at CCR5 and things like that, and then pushing those through into the educational realm. So we don't just go and stop from a computer, but we actually make something that can be held in somebody's hand. And I have to say, we have actually been printing models and giving them to patients in our rare disease clinic in Grand Rapids here. It's actually one of the few times I've ever seen a patient cry because they were overwhelmed at having some resource that they could use to talk about it. And so it's been very moving from the clinical side, what you can do with these prints. And something that you guys can be thinking about, how do you help support the clinics around you and the genomics? This is something that is surprisingly really useful. And so we've expanded this quite a bit. I think now we're up to 150 three-dimensional printing coordinate files, and you can see how complex we've gotten. So this is a keychain, fits in your hand, that's composed of 30 proteins. There's DNA and the histone wrap, and there's a whole complex that regulates it in gene transcription that we printed for one of our collaborating labs for their student who just graduated, and it was like their big celebration gift. So it just goes to show you can go very complex or more simple, highlighting variants. It's an unlimited potential field to move forward in. The other field is a little bit less about the variants and protein and more about gene regulation and really starting to think about non-coding variants and how they may work. And so this is a professor who's currently on sabbatical in my group, Bill, who's from the Grand Rapids Community College here. And he's really been trying to push and optimize tutorials and training for RNA-seq workflows. So how do you download diverse data sets from NCBI and download those reads, whether it might be liver, if somebody has an interest in liver or cancer or something, all the way through building, better tutorials of how to process the data on our servers or his server, and all the way into testing hypotheses and saying, well, how is COVID patient blood different from controls? And this is actually real data that we're getting ready to publish here pretty soon, that he has been working on in about 15 medical students in processing data sets from COVID blood samples from our hospital. One of the areas that I'm actively looking for a group to take some lead on, if anybody is interested, is looking at more of the complex variants that are difficult to link to biology based on inheritance structure, but we know have huge importance. And we call these QTLs, quantitative trait loci, where variants can either impact expression of a gene in various tissues of the body. So that's going to be an eQTL, E for expression QTLs. And we know that there are thousands of variants in the genome that alter the expression of genes. The really cool one that has actually come out recently with much better data sets are what we call SQTLs, which stand for splicing QTLs. So these are variants that actually contribute to how any two individuals may actually splice genes up slightly different. And when you think about splicing biology, this is more of an impact on proteins than a simple subtle missense variant, right? So in this case of this variant that you can see, this is actually the strongest signal for an SQTL in the entire genome, which is in this CNIH4 gene. And you can see if you carry heterozygous this variant, you actually completely alter which exons you include in the gene. And so can we make sense of that? What's the mechanism of how this works? How does that connect to any biology? Looking at GWAS studies, what's the complex inheritance? This is an easy one. We know it's one variant that's altering the splicing, and we can go in and develop mechanisms for that. I worked on sabbatical this summer for a pharmaceutical company. This is the kind of stuff that is very hot in pharmaceutical and pharmacogenomics, and actually trying to make sense of the COVID genetics actually runs in this realm as well. One of the other projects that I wanted to highlight is how do we do some of this characterization work faster and quicker? We just actually got both state funding, private philanthropy funding, and a foundation grant all within a few weeks to get this project actually going and now funded, where we're actually taking kids from the NICU throughout the entire state of Michigan now and doing 2 to 3 day whole genome sequencing, partnering with Brady Children's Hospital out in San Diego. And then we just secured funding to do rapid characterization, so 1 to 2 week turnaround times at slowest, to be able to give them these educational materials and variants. And so I anticipate the need of many students to be able to be ready to do this fast and quick, where they've already done 2 or 3 variants working from ClinVar, that they know what they're doing, and I can distribute projects through them as well. And say, hey, we've got this one, can you get me some material in the next 2 or 3 days, if possible? And not just the variants itself, but all the way through the educational material platforms. We've been launching touchscreen tablets in the clinic where the proteins actually can be sent to them quicker. Instead of waiting a week or 2 for a 3D print to come off, we can get them a 3D file, in a minute through the internet. Now they have something they can show in clinic that day. And then some of our work that Adam Underwood from Walsh University, I know a couple of you guys have met Adam when he came down to Alabama a few years ago when I was still there, which we've been working on optimizing some workflows for viral proteins and bacterial proteins, which I think is the biggest initiative that as we start thinking about big projects we can all come together around, really could be something really cool. And I got into this actually through a simple variant characterization project. The clinic came with a case of a patient and said, hey, this is our patient's case. If you look at it, they essentially have what they thought was just a very simple infection that about 14 days later turned into all their organs shutting down, right? And that should sound somewhat familiar right now. This case was actually 2.5 years ago, but that sounds familiar right now because that's actually the time course of COVID, right? You get it, and it takes about 10-ish days before you start showing a lot of the severe aspects of the infection. And then all of a sudden you rapidly progress into either you're going to be okay and get through it, or you can go into these cytokine storm syndrome, hyperferritinemic sepsis that we call it, which is actually what that patient had. And we worked up the genetic variant in it. And while we were working up the variant, and actually Hudson Alpha was involved, IREP, Casey's on here, they stepped in and helped do some work on this to help us understand this patient. And we made sense of this variant, and it was a weird mechanism. But we actually realized the variant was a healthy 16-year-old, never showed anything before. They got Epstein-Barr virus, and it took this delayed time course because the virus itself turned on a dominant negative variant, this frameshift variant, that in the RNA data we actually couldn't see until they got the virus. And it turned it on, and that caused the immune system to go haywire. And that haywired nature is a genetic switch that we're actually really interested in COVID. And so we started asking questions, well, this patient looks just like a COVID patient that passes away or has severe outcomes. How do we make sense of that? And how do we start building better tools for COVID to understand which genetics may be at work? And so we built this team up of multiple schools, many on this call, where we were looking at every single protein of the SARS-CoV-2. And we built up a database of these proteins, running them through this workflow that you see time and time again that we use. Model protein, run simulations, look at the protein by itself, look at the protein with the other proteins that it interacts with, bring in evolution, look at how the sites are under conservation or unique. We've been bringing in a lot of genomic data from sequencing the virus itself. How has the virus changed over time? And really trying to focus in on how are these proteins working? And more so, this is a lot of work, right? We did this in about 2 months from start to end for project completion. And that really pushes in this idea of distributed workload, right? We've got teams of scientists here on projects that when something like this comes up, I can reach out to UNA and Hudson Alpha and Athens and say, hey, can we bring together some more resources? Can we get some students on here? You guys focus on this protein, we'll focus on this protein, and start building this together. And then bring in the human genetic side. So we got into really complex structures of the virus docking and going into the cells and screening how human variants in the ACE2 protein, which is the receptor for the virus, might impact it. And we built a computational screen that was the first out, and now there's probably about 20 or 30 other groups that have done this, but we beat them all to the punch. And we got this paper out and, in a good journal. We're now moving that forward. We're trying to say, well, what about all the other interaction partners for these proteins? How do they interact with the human components? What variants exist in these? How does that impact COVID pathology? Which patients are at risk for certain things? And that's a lot of where my group has pivoted over the last few months, continuing those initiatives. We still have a lot of work that can be done. We have an entire new project kickstarting on the papain-like protease. And I think there are still hundreds of projects in COVID and other viruses that we can explore with students through projects. And so hopefully I've left you guys some ideas of, how we can integrate some of this stuff together, and where maybe some future avenues we can start building some student projects on. And I think we don't just, focus this on undergrads. I know some of the schools in code, and I have very heavily been going towards medical students. I have about 20 medical students right now actively working on projects, of which I think there's 10 papers in review. So we're making progress in how this can move faster and quicker for science, but then scaling at multiple levels, and even all the way down into high school and junior high schools. So we've been kickstarting some projects with a couple of schools back in Ohio where I grew up, and I think there are so many ideas that can be put forward. And my goal as a group is to provide you guys support for, if a student has an idea and you have no idea how to answer it, right? Don't hesitate to email me and say, is there some way we can do this, right? And don't just preclude it to, hey, is this a modeling of a protein? If a student comes and says, I want to know this non-coding variant that keeps on popping up, or I really want to understand how this gene is regulated, or how this tree protein is involved in this tree infection. That's actually a project I have a student at the community college working on. There are so many projects that can be taken and built. And really also we can be thinking from bigger sides now of what about other viruses? What about influenza proteins? What about RSV? How do we make sense of those proteins just like we did here? So cool. So that is the extent of the slides I have.
Well, we'll wrap since I know you have to run. Appreciate your time and thank you to our guest. And I hope that you all enjoy the other students' posters and appreciate it again, Jeremy.
Well, thank you guys, and congratulations everybody on outstanding posters. I'll continue to go through them and maybe ask some questions for some of you guys.
All right. Goodbye, everyone.
Bye.